Assalamualaikum and hi everyone. I hope you are healthy and safe. What are you doing during this MCO time? Let me tell you my plan. I think I want to pet a fish, a glowfish. Have you ever seen a glowing fish? Let me tell you, glowfish is a transgenic organism that expresses fluorescent color when viewed under ultraviolet light. What? Transgenic organism? Yes, it is. Transgenic organism is the new trait to the fish which does not occur naturally in the species. The DNA has been altered through recombinant DNA. DNA recombinant is the insertion of different DNA fragments or gene of interest and paste it into other organisms by enzyme. Do you want to know more about glowfish? To make this process clearer, let's study how glowfish can glow as a result from recombinant DNA. Let's hear further explanation from my friends Sajida and Shahada.
Choosing rainbow shark as one as the glowfish species, it obtains their very thick glowing body color from the sea coral. Let's say I want the fish blue in color. How can the fish get the color? Through DNA recombination, the blue color of rainbow shark must go through a few steps. First step is the isolation of the DNA from organism. To obtain a fluorescent blue color of rainbow shark, scientists has isolated the gene of interest from sea coral called Montiflora efflorensis, which code for their fluorescence color. Next is E. coli plasmid is isolated from the host cell. Second step is cutting the DNA with restriction enzyme. The restriction enzyme will cut the specific scene sequences, producing blunt end or sticky end at the both of DNA fragments. Third, the gene of interest is inserted into the plasmid vector, which is in this case, the gene from the sea coral is inserted into the E. coli plasmid vector to create the recombinant molecules. And now, the gene is joined using the DNA ligase.
And now, let's move to the fourth step, which is transforming the recombinant DNA. The recombinant DNA is inserted into the E. coli as a host, and it will replicate to make copies of genes in a culture. The last step is selecting the successful culture. The successful growth of the bacteria in the culture are selected and isolated again. After a few series of processes, finally, the recombinant DNA is inserted into the rainbow shark genes by the microinjection. The gene inserted will give the ability of the fish body to express the cosmic blue fluorescent color and appear bright under the normal light.
Other than become aquarium fish, glowfish is actually designed as a bioindicator in detect water pollution. Fish appear bright under normal light and fluorescent under blue light. However, the fish produce does not harm to them. In fact, glowfish are more vulnerable to the predation compared to the wild species. Other than that, do you know that the ability to produce light can give animal competitive advantages? For example, deep sea squid will inject glowing mixtures to startle predator and hatch fish like producing organs to adjust reflections of their body as a mask. Other organisms that have ability to glow include plankton, coral, and global. There are many other benefits with the application of the DNA recombinant technology. This technology will help improve life, food quality, and livestock, and other development that can save net. We hope that this can increase your knowledge and curiosity about DNA recombinant technology and genetic engineering. That's all from us. Thank you, goodbye, and stay safe.